Welcome to this lesson on measurement techniques for accurate data collection. In the laboratory, the quality of your experimental results depends heavily on how precisely you measure quantities. Today, we'll explore three essential instruments: the analytical balance, the graduated cylinder, and the pipette. These tools form the foundation of quantitative work in chemistry and biology. Mastering their proper use will ensure your measurements are both accurate and reproducible. Before we dive into specific techniques, let's understand why measurement accuracy is so critical. Precise measurements are the foundation of reliable scientific data and reproducible experimental results. Even small errors in measurement can compound through calculations, leading to significantly incorrect conclusions. When you measure carefully and consistently, other scientists can replicate your work and build upon your findings. This reproducibility is what makes science trustworthy and progressive. The three instruments we'll focus on today each serve distinct purposes in the laboratory. The analytical balance measures mass with exceptional precision, often to 4 decimal places. The graduated cylinder measures liquid volumes, providing a reliable method for handling larger quantities. The pipette delivers precise volumes of liquid, especially useful for smaller amounts. Each tool has specific advantages, and knowing when and how to use each one is essential for efficient lab work. Let's start with the analytical balance. Before taking any measurement, you must calibrate the balance according to the manufacturer's instructions. Next, ensure the balance is level by checking the bubble indicator, adjusting the feet if necessary. Always close the draft shield during measurement, as even minor air currents can affect readings. Finally, tare your container before adding your sample. This zeroes out the container's weight, so you measure only the sample itself. These steps might seem tedious, but they're essential for accuracy. Several factors can introduce errors when using a balance. Air currents are a common culprit, which is why we use the draft shield. Temperature fluctuations can also cause problems, as they affect air density around the balance. Static electricity can attract or repel lightweight samples, causing false readings. To minimize these issues, work in a stable environment, allow samples to reach room temperature before weighing, and consider using an anti-static brush for problematic materials. Moving on to the graduated cylinder, proper technique here involves 4 key steps. First, always place the cylinder on a level surface. An angled surface will give you an incorrect reading. Second, position yourself so the measurement mark is at eye level. Reading from above or below introduces parallax error. Third, read the bottom of the meniscus, that curved surface of the liquid. Finally, choose a cylinder size appropriate for your volume. Using a 10 milliliter cylinder to measure 8 milliliters is far more accurate than using a 100 milliliter cylinder for the same volume. The meniscus deserves special attention because it's a common source of measurement error. This curved liquid surface forms due to surface tension and adhesion between the liquid and glass. For most aqueous solutions, the meniscus curves downward, and you always read at the lowest point of this curve. Some liquids, like mercury, form an upward meniscus, and in those cases you read the highest point. Consistency is key. Always use the same part of the meniscus for all your measurements. Pipettes come in several varieties, each designed for specific applications. Volumetric pipettes deliver a single, highly precise volume, making them ideal for preparing standard solutions. Graduated pipettes allow you to measure variable volumes, offering more flexibility. Micropipettes handle very small volumes measured in microliters, essential for molecular biology work. Serological pipettes are designed for transferring liquids and often include markings all the way to the tip. Understanding these differences helps you choose the right tool for your task. Micropipettes require careful technique to achieve accurate results. Start by selecting the correct tip size for your pipette model. Set your desired volume using the adjustment dial, being careful not to force it beyond its range. Before your actual measurement, pre-wet the tip by drawing up and expelling liquid once or twice. This equilibrates the tip with your solution. When drawing up your sample, use a smooth, controlled motion, pressing the plunger to the first stop only. Jerky movements or pressing too far can introduce air bubbles and errors. Let's recap the essential principles we've covered. Master proper calibration for your balance, ensuring it's level and protected from environmental factors. Always position yourself correctly when reading volumes, and remember to read the meniscus at eye level. Practice smooth, controlled techniques with pipettes, and always pre-wet your tips. These 3 instruments, when used correctly, form the backbone of accurate quantitative work in the laboratory. With practice, these techniques will become second nature, and your experimental results will be more reliable and reproducible. Thank you for watching.
If you like this video, hit that like button and don't forget to subscribe. Visit CodeLucky.com for more such useful content.